Okay, Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody, and hello, everyone. I'll deliver about vitamin K metabolism, and it's our agenda for today's learning. Okay, vitamin K is one of the fat-soluble vitamin, has the function, the maintenance of normal coagulation because the vitamin K is necessary for post-translational gamma carboxylation of specific glutamic acid residues in a small group of proteins with calcium binding properties. Or we can say that the GLA proteins are glutamic carboxylate, gamma carboxylate, glutamyl proteins. The vitamin K dependent coagulation protein are synthesized in the liver and comprise of factors 2 or prothrombin 7, 9, and 10, which have a hemostatic role because they are procoagulants that arrest and prevent bleeding, and also protein C and S, which have anticoagulant role because they inhibit the clotting process. And how can we acquire a vitamin K? Okay, we can acquire a vitamin K in the green leafy vegetable. In the green leafy vegetable, rich of vitamin K1. The vitamin K1 is other name piloquinone. For the vitamin K2, we can get menaquinone from the natto. The natto is the fermented food Japan, and we can get it from the animal derived product like a chicken, like a cow, beef, grilled beef, etc. And the third one, there is vitamin K3, has the other name menadione, and it is synthetic and it has the water soluble properties. And the vitamin K3 or menadione is the feed fortified for the animal feed, like the cow, a swine, a goat, and for the poultry. Okay, it is the illustration. The green vegetable is rich of the vitamin K1, and the products of fermented food and animal-based foods are rich vitamin K2. Piloquinone can transform to menaquinone or vitamin K2 because of the intestinal microorganism like bacteroids and lactococcus. The intestinal microorganism can convert piloquinone to be the menaquinone in the large intestine or distal colon and terminal ileum. Okay, for the function of the vitamin K is coenzyme for the gamma carboxylase enzyme. So the gamma carboxylase enzyme has the role play as the convert inactive uncarboxylated prothrombin to be the active carboxylated thrombin. Gamma carboxylase can work because of the role of the vitamin K. The activated vitamin K is the hydroquinone. Vitamin K hydroquinone is an active form from vitamin K, and it is a good effect for our body. Okay. After the vitamin K hydroquinone help the helping the gamma carboxylase, the activated vitamin K will transform to be the activated vitamin K is the vitamin K epoxide, and the vitamin K epoxide must be continue its work, must be continue its activity. So it must activation. It must do its activation via the epoxide reductase, the vitamin K epoxide, because of the enzyme, and the enzyme name is the vitamin K epoxide reductase, will convert vitamin K epoxide to be the vitamin K quinone, and we can acquire the vitamin K quinone from the dietary sources like the green leafy vegetable and dairy products and animal-based products. The vitamin K quinone will be activated by the quinone reductase. So that's why the vitamin K quinone to be the active form is the vitamin K hydroquinone. So the vitamin K hydroquinone can continue its activity as the coenzyme. So the next for the MK4 is the one of the menaquinone. Menaquinone 4 is the one of the vitamin K2 form. So the vitamin K2 form has many menaquinone form like the menaquinone of 4, 7, 9, 10, 11 to 13 in our body. 4 is shown the number of the long chain. So the higher of the number of the long of the chain of the menaquinone is the more lipophilic properties, and menaquinone 4 belongs to the short chain menaquinone, MK4 short chain, MK9, 10 to 13 is the long chain of menaquinone. And for the menaquinone 4 is the unique. What's why the MK4 unique? Because the piloquinone will be converted to the menaquinone without bacterial action, because the menaquinone 4 is converted from the piloquinone or vitamin K1 to be the menaquinone 4 without bacterial action. Meanwhile, for the menaquinone 7, 9, 10, or 11, 12, 13 enforce bacterial action to produce its menaquinone. Okay, and it's the pathway of metabolism of the vitamin K. This overview. Okay, the vitamin K will be solubilized and emulsified by the mixed micelle. So the vitamin K can be absorbed in the brush border of the enterocyte. And next, the vitamin K will be repackaged so it can form the chylomicrons. Vitamin K along with dietary fat, cholesterol, lipoproteins, apolipoproteins to uh, to uh, to be the chylomicron forms, and then these chylomicrons are secreted into lymphatic system, and it will circulate in the blood circulation to go to the liver uh, for the metabolism, and 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 from the liver will be transported to the other peripheral tissue or other tissue or other organelle cells that need vitamin K activity. So the vitamin K is rapidly metabolized and excreted. So that's why the vitamin K is relatively low blood levels and tissue stores compared to those of the other fat-soluble vitamins. Okay. Because of the vitamin K rapidly metabolized and excreted to the urine or fecal, so the vitamin K's concentration in the blood and tissue store is lower than those of the other fat-soluble vitamins, such as vitamin E, vitamin B, and also vitamin E. Okay, how can we acquire the sources of vitamin K? Okay, the number one is food. The first one, food. Food sources of piloquinone include vegetable, especially green leafy vegetable, vegetable oil, and some fruits. Meat, dairy foods, and eggs contain low level of piloquinone but modest amounts of menaquinone. Menaquinone-rich foods are those with a bacterial fermentation stage. Yes, you have to memorize it. For the menaquinone-rich foods, because of the bacterial action in the fermentation stage, like the Bacillus subtilis bacterioides, like um, lactobacillus, lactococcus, but not for yeast, for food, yeast for and fungi, because the yeast don't synthesize menaquinone. So that's why the Candida albicans Saccharomyces cerevisiae belongs to yeast, cannot synthesize menaquinones. In the typical diet of developed countries, nutritionally significant amounts of long-chain menaquinones have been found in animal livers and fermented foods such as cheese. Natto, a traditional Japanese food made from fermented soybean, and it has high amounts of menaquinones content even higher than a piloquinone content of green leafy vegetables. Animals synthesize MK4 from menadione. Menadione is a synthetic form of vitamin K that can be used in poultry and swine feed. MK4 may be synthesized in animal tissues from menadione supplied in animal feed. The absorption rate of the piloquinone in its free form is approximately about 80%, but its absorption rate from foods is significantly lower. And piloquinone in plant foods is tightly bound to chloroplast, so it is less bioavailable than that from oils or dietary supplements. Consuming a vegetable at the same time as some fat, like the virgin oil or palm oil, can improve piloquinone absorption from the vegetable, but the amount absorbed is still lower than that from oils. Limited research suggests that long-chain MKs like the MK9, 10, 11, 12, 13 may have higher absorption rates than piloquinone from green vegetables. and they are the examples of the vitamin K rich foods from animal and animal derived products and its plant sources. The second one, dietary supplements. Vitamin K is present in most multivitamin or multimineral supplements, typically at values less than 70% of the daily value. It's also available in dietary supplements containing only vitamin K or vitamin K combined with a few other nutrients, frequently calcium, magnesium, and or vitamin D. These supplements tend to have a wide range of vitamin K doses than a multivitamin or mineral supplements, with some providing 4,050 micrograms, about 5.63% of the daily value, or another very high amount. Several forms of vitamin K are used in dietary supplements, including vitamin K1 as piloquinone or phytonadione, uh, and phytonadione is a synthetic form of vitamin K1, and vitamin K2 as MK4 or MK7. Few data are available on the relative bioavailability of the various forms of the vitamin K supplements. And one study found that the bound phytonadione and MK7 supplements are well absorbed, but MK7 has a longer half-life. Menadione, which is sometimes called vitamin K3, is another synthetic form of vitamin K, and it is usually for the animal feeds. And next, the third one, importance of intestinal bacterial synthesis as a source of vitamin K. Intestinal microflora synthesize large amount of menaquinones, which are potentially available as a source of vitamin K. Quantitative measurements on different sites of the human intestine have demonstrated that most of these menaquinones are present in the distal colon. Major forms produced are MK10 and MK11 by bacteroids, MK8 by Enterobacter, MK7 by Veillonella, and MK6 by Eubacterium lentum. It is noteworthy that menaquinones with a fairly long chains, MK10 to 13, are known to be synthesized by members of the anaerobic genus Bacteroids and are found in large concentration in the intestinal tract but have not been detected in a significant amount in foods. The widespread presence of menaquinones 10 to 13 in human livers at high concentration therefore suggests that this form at least originates from intestinal synthesis. Okay, presence of menaquinone 10 to 13 in human livers at high concentration. So that's why these menaquinones originate from intestinal synthesis. It is commonly held that animals and humans obtain a significant fraction of their vitamin K requirement from direct absorption of menaquinones produced by microflora synthesis, but conclusive experimental evidence documenting the site and extent of absorption is singularly lacking. The most promising site of absorption is the terminal ileum, where there are some menaquinone producing bacteria as well as bile salts. But the balance of evidence suggests that the bioavailability of bacterial menaquinones is poor because they are the most perfectly bound to the bacteria cystoplasmic, membrane, and also because the large pore is present in the colon, which lacks bile salts for their solubilization.